Okay, good morning, everyone. I hope each one of you is able to see what is on my screen. My name is Bumba Patrick, and Senior 5. By now, I am sure that at least most of you must be around this topic, and that's why I decided to begin with transport. But with time, may I know the learners that are okay. So with time, you're going to be able to understand. So transport, we normally do transport in plants and animals, but we like so much starting with transport in plants. And as you can see on this side, this is a small animation that just introduces us to this topic. How do plants actually transport materials where they're supposed to be? You can see that this is like a room, and you can see these are minerals, and they are being ferried by something which I will have to tell you a little bit later. And you can see there is this kind of suction pump that is pulling the materials up somewhere. But then you can also see there are some storage areas where they are stored. Now, you may not understand this animation right now, but with time you're going to understand it. Okay. I think we can start. I hope each one of you is ready. For you to better understand transport in plants, it begins. Just look in your lower right corner. This is a plant of chili pepper. Open here a little bit. Choosing this plant, it's a dipole. What you're seeing here is a cross section of the transverse section of the dicot root. Now, this picture too is a transverse section of the monocot root. And then this is kind of a longitudinal section, this picture 3, longitudinal section of a dicot root. Now, actually, what you're seeing here is you just pick just a small tip of the root here, like that. It is what is being magnified here in picture 1 and 2. Just that one small tip. Because if you want to understand how materials get into the plant, then it begins from the soil and the root part. And this is what I am having here. Now, from your O level, you find that you've been drawing what we call tissue plans. And I'm going to help you kind of draw it for you so that you can remember from your O level knowledge for better understanding. Okay, let me use this space here. What we begin with is the outermost layer, that is the epidermis. You can see it is double layered, and then there are root hairs there attached to it. So what we do is that we can normally draw. That's a root hair. I'm trying to draw the epidermis. I'm trying to draw the epidermis. You can see it. I'm actually drawing a tissue plan right from the real drawing. Of course, it's for biology drawings, you don't need to be like use a ruler or a compass. No, we just use a freehand. Now that is the outermost layer of the epidermis. And I've already told that it is double layered, so I can draw. Very fast. Now that is our epidermis. Okay. Then from there we go to the cortex. Now this cortex part It runs from here up to here. Now the cortex is packed with parenchyma cells. What you're seeing here, these are all parenchyma cells. In the middle, they're all parenchyma cells. Prima from Roviri, you may mute. Prima from Roviri, you may mute. Yes, please. Thank you. What you're seeing here, these are cortex. It is the cortex cells is packed with parenchyma cells. They're just thin cells. Now, there's no way how we normally draw them. We just leave space for tissue plans. Then next, we go to the endodermis. Now, this is the brown layer that you're seeing here. That's the endodermis. So I can also draw the endodermis there. I can draw the endodermis. Okay, that's the endodermis. Then I can go to the pericycle inside that layer. That's the pericycle. I can now also draw the pericycle you see there. Now that's this. First, we have so far drawn the epidermis. The epidermis, the epidermis. And now we have drawn the endodermis and pericycle. Now inside the pericycle, this is where you're going to find the vesicular bundle. Now the vesicular bundle, when you hear the word vesicular bundle, I mean phloem and xylem. Now for the case of the dicot, the transverse section to the dicot root, the xylem is in form of a star. If you can see this, it's just that it is halfway, but it's supposed to be in form of a star. It's a form of a star shape. So I can draw the star shape. I can draw the star shape. I can draw the star shape like that. Okay. And then on the side, this is where we find the phloem. So I'm drawing the phloem. I'm drawing the phloem. And I'm drawing the phloem. Okay. I'm very much sure that you have seen this before from your O level. This is the transverse section through the dicot root. Transverse section to the dicot root. And this is it. Just a cut. You cut from here and then you draw. Now, what we are going to study is how actually water and mineral ions are able to move. They move, they're absorbed via the root hairs. They go through the cortical cells. They cross the endodermis. They enter the pericycle until they reach the xylem, and then they are moved up the plant. They are moved up the plant. Now, if you are to understand how these materials, the water and mineral salts are going to move in the plant, then you must first understand the anatomy, cross-section through the stem. We're of course using the dicot. We are going to first ignore the one for monocot. So for better understanding, we are now considering. So whenever, if you are to understand these processes well, you must be sure of this structure as we are going to keep on moving. Now, mineral transport of water and mineral salts is going to involve three different processes. One is going to be first uptake of water by the roots, because water must be taken up. And then secondly, the movement of water into the vascular tissue of the root. Remember, I've said one, we are looking at uptake of water by the root, and then secondly, movement of water into the vascular tissue. And remember, I've already told you, when I talk of the vascular tissue, I mean phloem and xylem. But for our case, since we are looking at uptake of water and minerals in the roots, we are going to concentrate on the xylem, because it is the xylem which is in charge of transporting actually water up the plant. Then after water has been moved into the vascular tissue, that is phloem and xylem, then how does that water ascend That is movement of water up the stem. Movement of water up the stem. As I've told you already, if you can look at this picture, so we are going to begin with uptake first, okay? And then how water is going to be taken up, absorbed by the root hairs. Then when it reaches here on the endodermis, after the water has been taken up, how does it enter the vascular tissue, that is the xylem? That is the second bit. Then after water has reached the xylem, how does it go up? How does it go up the plant? Seeing techno F1, you have a question. Your hand is up. Maybe for purposes of understanding you better, next time you should use your name. You can tell me your name and you ask the question. Your hand is up. Yeah, the one who is using Techno F1, your hand is up. Can you please? And also the one using Techno Spark 2, you can mute. You can mute so that we can. Okay. Maybe you don't have a question. I can continue. The one using, please, the one using TechnoSpark 2, there is an option on your Zoom where you can mute. The one using TechnoSpark 2, there is an option where you can mute so that we don't get interrupted by the background information. The one using TechnoSpark 2, please mute. Okay. Okay, so I've already told you that we are going to begin with this first objective, that is uptake of water by the roots. How is water taken up by the roots? Uptake of water by the roots, of course, going to begin with the root hairs. One, normally plant has numerous root hairs. Now, those root hairs are going to penetrate the soil particles and they're going to absorb water from the spaces. You can see this is again a transverse section through the dicot root. And what I'm calling the root hair, it is this one here. This is the root hair cell. From your O level, we have studied the root hair cell. So this is one root hair cell. This is another. This is another. There can be many. Normally, there are many to increase the rate of absorption. They increase the surface area for absorption of water and mineral salts. And there are quite many. They are very thin, one cell thick. Now, those root hairs are the ones that normally absorb water, and water is drawn into the epidermal cells. This is the outer layer, which I'm calling the epidermis. Okay, it's the outer layer, which I'm calling the epidermis. Now, you're going to find that you have soil particles around. We're actually inside the root. We're inside the soil. And you're going to find that you're going to be having water. Let me use just this blue. This is representing water. You can see we are having water molecules around in the soil. So I'm only doing it for one root hair, but remember, this is happening for all these other root hairs. It's happening for all these other root hairs. And water, this is going to be the path. Water is going to be absorbed by the root hairs, and then it should go through the epidermis, then the cortical cells, then endodermis, pericycle, up to the xylem, and then the water must move up. Okay, and then water must move up. So water is drawn into the epidermal cells by the process called osmosis, because osmosis, it's a movement of water molecules from a region of high water potential to a region of low water potential. or you can look at it in terms of concentration, from where there is a low concentration of solute, and that is a solution with low concentration of solute normally has more water than the solute particles. Now, normally what makes up the solutes in the root hair or the sap of the root hair cells are going to be sugars, as well as some other metabolites like ions, okay? And then which lowers the water potential. Now, what do I mean by water potential? With water potential, I mean by the tendency or a bit of the water to leave a system. So normally, by conversion, normally, pure has water potential of 0. Pure, if I'm assuming if this was pure water, so its ability to move the system, we normally say that it has a 0 water potential. It has that is the highest water potential. Now, when you add a solute, normally these water molecules can move freely very fast. When you add a solute like sugar, okay, into it, now this sugar is putting a drag effect on the water molecules. So these water molecules are not going to be moving freely or with ease. So when you add a solute into water, you are actually lowering the water potential. That is its ability to move freely. So you find that in the root hair, you normally have solutes, which solutes normally come are translocated via the phloem. Normally, this phloem around around this xylem. So they have sugars inside. So now it is those sugars that lower the water potential, its ability to move. So when the water potential is lowered of the cell sap than the surrounding soil water. Now soil water here is going to be in the soil solution here, you're going to have a very high water potential, like its water has a higher ability to move. Now inside, there are some people who need to mute. Please, Daniel, I'm seeing Daniel, I'm seeing Techno F1. You have an option on your Zoom to mute. You have an option to mute yes, Daniel has muted, but Techno F1 has failed to mute. Your background sound is interrupting our lesson. Morris, you can mute also. I'm seeing Morris and Morris, please mute. You have an option of muting your background noise. Okay. When you enter the lesson, please mute. There is that sign of a microphone. You can first mute. Thank you. Okay. I was still telling you that this water that is surrounding the root hair has a very high water potential compared to the inner, to the sap, the solution in the sap of the root hair cell. Now, what happens is that water always wants to move from where there is a very high water potential to where it is at a low water potential. It's like where are they saying that I'm low, so it wants to go there. So water is going to diffuse from the soil solution, and then it's going to the root hair cells at a low water potential, okay, via what we call a concentration gradient. And that process of movement of water from a high water potential to a low water potential is what we term osmosis, as osmosis. So water molecules then pass across the cellular cell wall and the protoplast. Now, the cellular cell wall, what you must know is that for any plant cell, generally, a plant cell will have a cell wall. A plant cell will have a cell wall, and then it will have a cell membrane. It will have a cell membrane, and then a vacuole wall. A vacuole wall, like that. Now, in between the cell membrane and the cell wall, this is where you find the living contents of the cell, which include the cytoplasm. Okay, if I can get some blue pencil here, which may include this cytoplasm, There are some cell organelles like the mitochondria. You'll have a nucleus here. You'll have chloroplasts inside. Those are what we call protoplasts. Now, protoplasts, this word protoplast means the living contents within the cell. The living contents within the cell. And normally there are all those organelles. For some of you have forgotten, a cell organelle is a distinct part of a cell that performs a particular function. I repeat, a distinct part of a cell that performs a particular function. So whatever is surrounded by this cell membrane, we call it, they are all part of what we call a protoplast, or the living contents of the cell. Okay, so into the subvacuole. So the water then passes to the vascular tissue. So the vascular tissue, I mean the phloem and xylem. Now that's in summary. I'm actually summarizing uptake of water by the roots. Begins with the root hairs. They are numerous to increase the surface area for absorption. So normally, the soil water solution surrounding the root hair has a high water potential compared to the sap solution. So water moves by osmosis from the soil solution into the sap of the cell, okay, by a process called osmosis via the epidermis. That is the summary of water uptake. Now, something about the cell wall. The cell wall is freely permeable. I don't know whether you've seen people who put on shoes which we call Crocs. There is a different kind of shoe which is trending now. Now, those Crocs normally have those powers in them. That's how a cell wall is. It is freely permeable. Things can just enter freely and anyhow. It is freely permeable. But for the case of the cell membrane, it is freely permeable. Not everything can just move out. That one, you must know it. Now, if we can just examine just a section of the root hair with the cortical cells up to the endodermis. Okay, wait a bit. Let me first rub off this. It may confuse some of you. And okay. So if we can just zoom, okay. Someone has a question. Okay, Gerard saying good morning. Yeah, Gerard, I think you came a little bit late, but I hope you are able to follow. So if you can just zoom, what I was saying, that just this section here, is what I'm having here. What you should not forget is the root hair, which is part of the epidermis. From the epidermis, we go to the cortex. From the cortex, we go to the endodermis. This is the endodermis. From the endodermis, pericycle, and then into the xylem. For some of you may not get it in this angle. Let me also put for you another one here, so that you can actually understand it better. Okay. So that you can understand it better. Let me get the pen. Yeah. Okay. So when we are to zoom into just this part, because to understand uptake of water, you must understand the anatomy. Okay. So we begin with this outer layer, which is the epidermis, where even the root hairs are part of the epidermis. Then next you have the cortex or cortical cells. The cortex is packed with parenchyma cells, packing tissue. Then from there we go to the endodermis, then pericycle, and then we can go to the xylem, the vascular tissue. Now, what you must know, there is some special feature about the endodermis. Are you seeing those kind of purple things inside between the cell walls. This is called the Casparian strip. It's called the Casparian strip. Yeah, is saying, may I get, okay, we are first doing the lesson. And then here, we are also having another Casparian strip here and another Casparian strip here. I think you can be able to see it. Okay. So once you understand this anatomy, you will never and never fail to understand this thing. Anyhow, so water is going to take two ways. One is this route, which is being represented by this red or maroon line, which we call the apoplast pathway. Now, the pathway in between the spaces all. I've already told that the cell wall is freely permeable, so the water is just going to be going through those spaces like that, like that, like that. So we'll have to go through those spaces like that until it will reach here, the endodermis, which we are calling the Casparian strip. And I'll tell you what it does, okay? Then the other pathway is, okay, wait. The other pathway is via the simplest. Now, this simplest pathway is the pathway through the cytoplasm. Oh, the cytoplasm. And you can see being represented by this blue line. It goes slowly, it goes via the cytoplasm, and then up to when it reaches the xylem. Now, there are two differences. We're having route 1, and we are also having route 2. Okay. Now, check for route 2, that is the simplest pathway. Water is actually going up to straight up to the xylem. But check for route 1, water is being blocked. Water is being blocked at this point, the Casparian strip. The Casparian strip. Okay. Now, what happens is that because the cell wall is freely permeable, so water is going to move, and actually up to 90% of the water goes via this pathway. Okay, via the apoplast pathway that is in between the spaces of the cell wall until it is blocked here. Now, this water must be diverted to go through the simplest pathway because there are very many reasons, actually, which I'll talk about. There are very many reasons as to why the water is blocked via the Casparian pathway. Okay, and there are some questions. I don't know whether you guys, if you have understood, let me, let's try to answer some of these questions. Question 1 says that the process by which water enters the roots due to diffusion is termed as the process. You can raise your hand and you tell us the answer. The process by which water enters the roots due to diffusion is termed as A, we are having endocytosis. B, we are having active absorption. C, we are having osmosis. And then D, we are having passive absorption. Yes, Daniel?
It's by osmosis.
Okay, thank you very much. Actually, the best answer is actually osmosis. Okay. And I already told that with osmosis, if you can still remember, water is moving from where it is at a high water potential to where it's at a low water potential. Okay, thank you very much, Daniel. Then question 2 says water potential of the root hairs cells is, A says equal to soil water. B says more negative than that of soil water. And then C says more positive in comparison to soil water. And then D says none of the above. And then D says none of the above. Uh-huh. What do you think? Anyone? Anyone? Okay, maybe you've not yet gotten this concept well. I could revisit it a little bit. I've already told you that, okay, let me use this. Yeah, I say that by convention, water potential is given the value of 0. Okay, now if you're having a system, using this beaker to represent a system, and we're having water. Okay, now this water has it has an ability to move out, to flow freely. If you put a membrane here, it can be able to move. That is a semi-permeable membrane. It can be able to move freely out of this system. You see that? Now the thing is, once you put a solute here, a solute can be like sugar or salt. Now the sugar molecules are going to reduce the movement of or the mobility of the water molecules here. And so they are lowering the water potential. That is its ability to move or flow out of the system. Okay, that is when you have added a solute, which means that normally a solution with more solute has a low water potential. And let me put, let me just get a blue pen here. I'm assuming these blue dots are representing a solute. They are representing a solute. So normally, a solution with more solute has a low water potential than a pure solution, because a pure solution doesn't have water solution doesn't have solute, so its molecules can move very fast and freely because nothing is hindering it, nothing is obstructing it in its movement. So a pure solution has its water potential high, but then a concentrated solution has its water solution low, that is water potential low. Okay, so normally in the root hair cell, because the root hairs are the ones that take up the solute, the root hairs are going to contain sugars in them, which sugars are synthesized in the leaves and they are stored either there. Than the inner sub solution. You'll excuse me for that. Okay. So that means the outer solution has high water potential compared to the sugar solution in the root hair cell. I hope you guys have understood that. And so the question was saying, water potential of the root hair cells is 1 equal to cell water. 2, it was saying that negative than that of cell water. And then 3, more positive in comparison to cell water, and then none of the above. What do you think is the answer? Can I have someone? Can Can I have someone? Daniel gave us this first answer, but then who is going to give us this next one? Okay. Okay. The answer is question says water potential of the root hair cells is more negative than that of cell water. Why more negative? Because it has more solute concentration in it than that of cell water. So that's why it has more negative. That is what it is more negative. Because by convention, pure water has that pure water, that is water without any solute in it. It has zero water potential. So when you add a solute, like sugar solutes, then it becomes more negative. So that's the answer. Then number 3 says, water will be absorbed by the root hairs when the external medium is, water will be absorbed by the root hairs when the external medium is. So you find that water is going to be absorbed by the root hairs when the external medium is hypotonic. What does hypotonic mean? Hypotonic means a less concentrated solution. It's going to be less concentrated, less concentrated solution. And then hyper means a more concentrated solution. More concentrated solution. And then iso means iso like same concentration. Going to be same concentration. And then means something thick, a thick solution. It means something thick. Now, when they say that water will be absorbed by the root hairs when the external medium, now that is, if you can take an example of the root hair cell, and I'm assuming you are having soil particles around it. You're having, these are soil particles. You're having soil particles around, having soil particles around the root hair, and then those soil particles have water, and they have water molecules. They have, I'm assuming they have water molecules. Now, for this water to begin moving inside, it means this external solution, which is the water, must be less concentrated. It must be concentrated so that it can have a high water potential. If a solution is less concentrated, okay, less concentrated means, let me add it here, less concentrated means high water potential. Less concentrated means high water potential. Less concentrated means high water potential. Okay, so for water to be absorbed, to absorb means to be taken up by the root hair like that, so that it can go into the xylem. It means that the solution must be less concentrated. So the answer is C. Okay, and then active absorption of water by roots from the soil is active absorption of water by roots from the soil is affected by. With active absorption, it means that energy is being used. Okay, the active absorption means that energy is being used, which energy normally is in form of okay, if you are online, please you try to mute. You try to mute, because if you don't mute, your background knowledge is like disturbing us. So you try to mute. Okay. Thank you. Thank you. So I was still telling you that active absorption means something active. It means that energy is being used. A lot of energy is being used, and which energy is in form of ATP. Okay. So for active absorption to be used, it means so active absorption of water by roots from the soil is mainly affected by essays also. Okay. Osmotic uptake or osmotic uptake. Oh, sorry, osmotic concentration of cell sap. They're having tension in cell sap due to transpiration, and then sucking power of the root hairs, and then typical tissue organization. I don't know whether any one of you has seen the answer. I don't know whether any one of you has seen the answer. Active absorption. A says osmotic concentration of cell sap. Will this one have? Then B says in cell sap due to transpiration. Now, what you must know with the tension, that is, it's a force which tends to pull two poles apart. Then with sucking power of the root hairs, the sucking power normally can be due to concentration, and then typical tissue organization. This is obviously not part. So we should choose from either A, B, or C. Okay, you guessed it right. I don't know whether you, so you find that we may go with A. And why? Because with the osmotic concentration, especially for active absorption normally comes about due to due to difference in concentration. Now, for molecules to be absorbed by active transport, it normally occurs as a result of one side having less, but you want to move molecules to that side. So that's when active absorption comes in. The questions about the other slide says absorption of water through roots can be increased by, it says increased transpiration, that is loss of water molecules at root surface, and then increased rate of photosynthesis, and then decreased transpiration, and then decreased absorption. Now, I may first skip this question. It will come in a little bit at another slide. Then question 6 says that when the concentration of soil solids is low, the absorption of water is, does it retard? Does it increase? Does it remain the same or stops completely? When concentration of soil solids, remember, okay, let me first remove this. Remember, I said that when concentration of soil solids is low, that means that solution has a high water potential. So if it has a high water potential, absorption of water is going to increase. It's going to increase. Okay. Then lastly, as a summary, we are having there are about 3 routes by which water moves through the parenchyma tissue of plants. One will be the apoplast pathway. And then secondly, it will be the simplest pathway, which you have already looked at. And then thirdly, it will be the vacuolar pathway. Okay. Now the apoplast pathway is that pathway which goes through the non-living parts of the cell. That is these spaces being represented by these purple lines. Now, this is the apoplast pathway. Okay, and then two, the simplest pathway. This is the pathway that goes via the cytoplasm. Okay. Now remember, I told that the protoplast. Okay, let me write it here for some of you who may have forgotten. The protoplast, and those are the contents which are normally surrounded by the cell membrane. Okay, going to be it goes through the living parts of the cytoplasm of the cell, as is in the cytoplasm, and the cytoplasm of the neighboring cells connect to plasmodesmata. These are the plasmodesmata that you see, those interconnections. And then the third one, as water is moving, is going to be the vacuolar pathway. Now, the vacuolar pathway here, I will have to include the vacuole wall. What you are seeing here, this is the outer one, is the cell wall. This is going to be the cell wall. And then the next one is going to be the cell membrane. Please, when you are online, please mute. Mulonji Faith, please mute. Mulonji Faith, you can mute. This is going to be, sorry for that, this is going to be membrane. Let me first clear off this. Okay, let me get a blue pen here. Let me get a blue pen. Okay, this is going to be the cell membrane. It's going to be the cell membrane. And then I'm drawing the tonoplast which surrounds the vacuole wall. So inside here you're going to have the vacuole. Now that is the pathway which is vacuolar. So it actually looks like very much similar to the simplest, but it's only that in this case water, let me just use in this case water will also go through the vacuole, then the cytoplasm, and then the vacuole. If I can draw more vacuoles here, like that, and it will go like that until it reach the xylem. And you can see that here you have the Casparian strip, which I'll talk much more about. I'll talk much about it in the next lesson. You can see this is the Casparian strip, and the arrangement still stands from the epidermis, then the cortex cells, and then the endodermis, and then the pericycle. That is always the route through which this water is going to continuously move. So at this moment, I'm seeing we are left with about 5 minutes. We're left with about 5 minutes, and then we end this lesson. So do you have any questions? I'm leaving these 5 minutes just to answer, and then just anything that you would wish, or if you have any comment, which you can raise your hand and you tell me. Yes, any comments from the audience? Any comments about the lesson? Just raise the hand and then you tell us. Or you could even have a suggestion that maybe after this topic, you can take on this like that. So I'm saying some people have gone off maybe because of data. Some people probably have gone off because of data. So any, do you have any suggestion, any comment before I wind up? Okay, maybe For now, maybe you don't have any comment, but maybe let me give you a summary of what we have just looked at. We have, for today's lesson, in summary, we have looked at the tissue plan of the transverse section of the dicot, and we have seen the anatomy of how the water is actually going to be taken up. Okay, and we have said that water is going to be taken up if the outer soil solution has a high water potential, the cell subsoil taken up by osmosis, and the root is going to go via the root hair cells, then epidermis, and then cortex cells, and then endodermis, okay, and then recycle up to the xylem, and it's at the xylem that it will be taken up the end. And we have also said that in the endodermis, there is a layer which we call a Casparian strip. And that Casparian strip prevents water that is entering via the apoplast pathway to continue, and then it is diverted to take on the simplest pathway. Okay, and when it takes on the simplest pathway, that is the pathway through the protoplast or protoplasm. Now, the protoplasm means the living contents within the cell. So for today, we have basically put emphasis on just uptake of water by the roots. So in the next lesson, we shall be able to look at movement of water into the vascular tissue of the xylem. That is, when water has reached the endodermis, then how does it cross from the ends to enter the vascular tissue, that is the xylem? That will be our next lesson. Then after that, we shall look at movement of water up the stem. Now, that is when water has reached the xylem. How does it move up? So if you have no question, I would like to end this lesson here. Do you have any question? Okay, thank you for being active learners. I think Daniel was even able to answer some question. And I'm stopping here. Enjoy and goodbye.
Thank you, teacher, for teaching. That was a good check. You have left? Okay, so the class has ended. The teacher will share the slides in our group. Have a good day.